Okay, I will be discussing now today because the other day where I left, I left that the nerve terminal. I made the diagram of the nerve terminal and how the nerve communicates to the other nerve. That was our question. That means in the last class I left that the transmission of impulse from one nerve to another nerve, how it happens. That was the question. Today I will be talking on that aspect. So that means the transmission of one nerve to another nerve happens by a contact with the other nerve. This contact may be of different types. This contact may be an electrical type or a chemical type, which I would be dealing with. Today I will be dealing with some part of it. So here I have made a synapse by my own, and you can just see that. I will explain the other aspect. Overall, this particular topic I want to deal in two parts. The first part, I want to give you an idea about the definition, then types of synapses based on the nature of transmission, and the structural parts of the synapse, the functions of the synapse, the morphological types of synapse, and events during synaptic transmission. That means listing the various events during synaptic transmission. These 6 topics I would cover today. The remaining part, that means the vesicular transport, the neurotransmitters, the properties of synapse, and the clinical aspects of these things I will be dealing with my next class on Wednesday. Again, my classes would be in the same time frame. Move on. What is the definition of a synapse? Before that, what is the meaning of a synapse? The synapse is derived from the word, a Greek word, synapsis. The meaning of this is conjunction, joining together, which in turn has derived from together. The conjunction has been derived from together. And so then another thing to fasten to, that means just to try to make it contact. It's also known as a clasp. Clasping means trying to catch, grasp. So that this is the meaning of a synapse if we are talking in terms of the origin of the word. Now, if I were to now bring this thing in a definition form, I just put this thing in a simple words. This is the junction between two neurons is called a synapse. Okay, it's too simple a definition. Maybe the next part would be a better definition. If you are seeing, it is a specialized junction where transmission of information takes place between nerve and another nerve, or muscle or gland cell. So that means that completes the overall definition of a synapse. That means it's a specialized junction which transmits the impulses to the nerve, to another nerve, or to a muscle, or to a gland. So this is a more comprehensive definition, but when you can use the first one also, there's no doubt about it. One thing we should want to consider, it is not an anatomical continuity, because there are no single nerve reaching to the brain, because the synapse, what happens, some nerve end in at one portion, and from there a new neuron begins, and this neuron reaches to a particular part in the brain or a brain stem, and then from there another set of neurons. Like that, there would be more than 2 or 3 connections before the impulse reaches to the or it has to pass through the 2 or 3 connections, minimum 3 connections if you are talking about sensory information. Minimum, I'm telling. So then it reaches to the brain. So bringing about the action again, that would have another 3 to 5 connections operating upon to bring about. Maybe here there may be a definite 2 connections are there. Now here in the top I have made the synapse here. The synapse here I have just made this ball and socket. So this ball and the socket, this is the socket represents the postsynaptic site. The ball represents the presynaptic terminal or a presynaptic proteome. So this is what the simplistic diagram of a synapse. Maybe you can try and try to make it in your exercise books. Okay, moving on, there are, according to the nature of transmission, because the nature of transmission in the synapse, in here in this thing, whether this transmission is electrical or chemical. Each has an advantage, each has certain important aspects. So it happens by 2 main things, that is electrical transmission and chemical transmission. In the chemical transmission, it is exactly a chemical, it's a transduction. The synapse acts as a transducer. So that means an electrical impulse is converted to chemical activity, chemical energy. From the chemical energy, an electrical energy is generated. So that means there are 2 transducers in series or placed. So that is a chemical synapse. In case of electrical synapse, there is no change in the energy pattern. So that means electrical energy is transferred to another nerve through an electrical energy. There is no transduction. So that means it is a no transduction here, a transduction here. That is the theme of the nature of these things. Putting it in the simpler terms, electrical synapse and a chemical synapse. Here is a diagram of an electrical synapse. Here there are membranes of the 2 cells actually touch and they share proteins. Can you see here in this box? You can just see that this is a presynaptic terminal and this is the postsynaptic sites. Sorry about that. So this is the presynaptic terminal. This is the postsynaptic site. You can see these junctions, these cell junctions, this cell and this cell, these cell junctions. This is amplified here, and once you amplify, this is having a sort of a gap junctions wherein they allow certain molecules to pass through easily. So now through these gap junctions, whenever the electrical activity comes here, the molecules pass, ions pass through. The action potential arriving here will produce a depolarization in the next membrane, and that is how the electrical impulse is transmitted. So here I have written, in these membranes, 2 cells actually touch. They share the common membrane, allows the action potential to pass directly from one membrane to the another membrane here. The importance or the function of these things is a fast transfer of the signal without any delay. However, this type of synapses are not present all through our body, because these type of synapses have their limitations, because their main limitation is they are bidirectional. And the second limitation is the signal strength will diminish in relation to the passage. So that means to excite the postsynaptic membrane of this, you require a greater input to this, because there is a loss of a signal strength. For that reason, that is another important thing. So it is bidirectional. That means it may come back to this thing. So that means that's another important disadvantage for this. where they are present. If you are asking this question, are they present in human beings? Yes, certainly in heart they are present. What the intercalated discs in the heart. So these intercalated discs in the heart, they transfer the electrical signal from one cardiomyocyte to the other one easily. Especially the atria and the ventricles, they beat in a synchronized manner. So because of these electrical synapses. They are present in our retina, that is in the eye, wherein the signals are off centers and on centers. They work in together. They are also present in certain areas of the brain and brainstem, where there is a pacemaking activity is required. Just like our respiratory pacemaker, the respiratory activity is generated in a ramp signal. So this pacemaking activity, you require a synchronization of the events. So that is where they are present in the medulla. Especially where there is a synchronization. Synchronization means a collective action of a group of neurons or a group of muscle cells. Then you need electrical synapse. Pacemaking, yes, generation of the activity is it's required. This is about electrical synapse. Then coming to the chemical synapse, in a chemical synapse, I have already mentioned electrical activity is transferred to a chemical activity. That means a transmitter is released, and this chemical activity is converted to electrical activity. In a chemical synapse, electrical activity in the presynaptic neuron is converted to, in the form of a release of a chemical, what is called a neurotransmitter. And this neurotransmitter binds with the receptors, and that receptor activates the postsynaptic or produces an electrical signal in the postsynaptic membrane. That is a chemical synapse. Let us see the these different parts of the synapse. If you are looking at these things, I have taken the same diagram. I have just put these parts of the synapse. You have what's this is the presynaptic terminal. That is the presynaptic terminal. So this is the postsynaptic terminal. It's the electrical synapse. You can just see here, it is also a presynaptic terminal. This is a presynaptic terminal, and this is a postsynaptic membrane. So that means the parts, there are mainly three major parts. One, presynaptic. This is the presynaptic knob. This is the presynaptic terminal. And then we have the synaptic cleft. So that is the space here. Especially this is there in case of a chemical synapse. It is not there in a electrical synapse, because electrical synapse, these membranes are coming in opposition to each other. So then there is a postsynaptic membrane. This is a postsynaptic membrane. Those three are the components: the presynaptic, synaptic, and postsynaptic. So this is the myelin sheath, where the presynaptic these things is happening. So that is what I have drawn. So this is the synapse happening with the other neuronal part, either maybe axon, dendrite, or soma. Okay, that is what the parts of the synapse. Now, having been considered, we have three parts mainly: presynaptic component and the synaptic space and the postsynaptic component. That is what I have written here for electrical synapse. For the electrical synapse, I have mentioned this is the presynaptic component. And what are the constituents or the parts in the presynaptic terminal? You see mitochondria. You see mitochondria there. You see gap junctions between this membrane and this membrane. That's a requirement. And there should be a free flow of ions. These gap junctions should allow the free flow of ions. Of course, you require energy level, which is coming from the mitochondria. And other components of maintaining these or sustaining these gap junctions, so whatever the translational aspects, they should be present here. In case of a postsynaptic membrane, this side, you require ion channels. Of course, This is the directionality of the ion channels. So then mitochondria, because to keep the ion channels or the membrane in a polarized state, you require energy, and other components as required for the presynaptic terminal. So this is precisely the parts of the electrical synapse. If you are looking at a chemical synapse, it's more complex. So this one have a presynaptic component, postsynaptic component, and a synaptic cleft. Now, if you are looking at the presynaptic, this is the presynaptic component here. This is the presynaptic component. I have taken it from a genome there. So now this presynaptic component is the ending that contains neurotransmitters, mitochondria, and other cell organelles. So this I will be coming one by one. The postsynaptic component, this is the postsynaptic component, wherein you have the neurotransmitter receptors, neurotransmitter receptors to attach with the neurotransmitters. And synaptic cleft is the space between the presynaptic terminal and the postsynaptic terminal. It would be around 20 nanometers wide, and definitely it would have a delay because the impulse has to be transferred. There would be approximately 0.5 millisecond is the transmission delay. So that means it takes the quantum defined amount of time for that transmission. Now, moving on, the presynaptic parts. Now I am going in detail with the presynaptic parts. What are the things to be, or what are the things required in the presynaptic terminal? One, the myelin sheath, which was, suppose this is the myelin sheath was lost. So if I were to go back here, if you are looking at here, I have made a myelin sheath here, and this myelin sheath is not there. So that means there is no, it is not covering the myelin sheath. So that means you should have the sodium channels in abundance. That is the one component. The second thing is, if you are showing the synaptic button, so that means there is a widening or flattening of the terminal. So that means that is to increase the surface area of contact with the postsynaptic components. So, or fitting into the particular modality or particular thing. So these things are required. This is a widening of the terminal or flattening of the terminal, what are called synaptic buttons. So then in the synaptic buttons, where they make a contact with the postsynaptic site, or where they have a vicinity of the postsynaptic site, so there are what are called active zones. These are active zones. These active regions are nothing but the collection of voltage-gated calcium channels. There is a high-density voltage-gated calcium channels out there. So this is one, and another protein, that is the protein which is necessary to hold on to these vesicles. So these are T-snare proteins. I will talk about T-snare proteins. If you are looking at here, so these are this is the synaptic vesicle. If you are looking at this is synaptic vesicle there, and this is the membrane. That means the postsynaptic membrane. So this is the synaptic vesicle and the synaptic membrane. This is synaptic membrane here. This is coming. So when this vesicle comes here, we have in this membrane, we should have the calcium channels on one side, and the syntaxin and the SNAP25. The syntaxin and SNAP25, especially syntaxin, is known as a T-snare protein. So now, in addition, this presynaptic terminal will have a large density of voltage-gated calcium channels, especially the calcium channels, because calcium is very much important for the transmission of these, or for the activation of these vesicles. So then to bring the energy, you require a large number of mitochondria. That's one of the component. So now to transfer whatever the material, or even to transfer these vesicles, we require microtubules. These are intracellular cytoskeletal structures, and these cytoskeletal structures, they would move the vesicles from one place to the other place. So that means vesicular transportation, docking, release, and again getting back is requiring these microtubules. Then we require in certain, there are other group of proteins here, because this I have this, the genome, it has not been marked. Maybe I am just typing. So here you require additional protein. These are called transporters or neurotransmitter transporter. So they are similar to the glucose transporters, what we call as a glucose transporters. So these transporters are essential because most of the neurotransmitters or one neurotransmitter released is uptaken or taken up by the presynaptic terminal. And when it is taken up by the presynaptic terminal, that requires a transporters. You have a norepinephrine, serotonergic transporters, dopaminergic transporters, glutamate transporters. So these all transporters are present on this presynaptic terminal, so that the minute the transmitter is released, that would be taken back by the presynaptic terminal. That is one thing. Sometimes what happens, these two synapses are guarded or welded by the glia or astrocytes. And when they are welded by the astrocytes are enriched with these transporters, but these astrocytes would provide the whatever the material. Say for example, glutamate is taken up by the glutamate transporters, and now the glutamine is transported to the presynaptic terminal, and then for the glutamine to glutamate synthesis. So that is how the astrocytes also try to work on. That means astrocytes are guarding together here. So you have the astrocytes here in the neurons. Now, so we have in the presynaptic terminal, we should have an enzyme system. I said the glutamine has to be again made to a glutamate and packed in the Golgi complex and the packaging material. So that is one thing. So now in case of a acetylcholine, what happens? Acetylcholine is released here. Now this acetylcholine released is catabolized or destroyed by the choline esterase, an enzyme which makes it as an acetate and choline. Now these acetate and choline levels in these extracellular sites, they increase. Now these acetate transporters are there. So you require a transporters. Acetate and the cholines are transported inside, and once they are transported inside, so that would be put them together. To put acetate and choline together, we have what is this choline acetyl transferase enzyme, CHAT. The choline acetyl transferase enzyme, CHAT, is required. So these joining molecules, the enzyme systems for the synthesis or assembling of the raw materials is there in these. Here I have not shown. Then we have two type of vesicles. The vesicles which are clear, here we don't see them here. We all have the dense vesicles. The clear vesicles we have that contains the simple molecules. That means low molecular weight molecules, justifiably acetylcholine or a single amino acid, glutamate, or something like that. So dense vesicles, they contain the peptides. The peptides may be the substance P, bradykinin, or any other enkephalines. These peptide neurotransmitters, they have a dense vesicles. So it does not matter whether it is a clear vesicles or dense vesicles. They contain the neurotransmitter. The neurotransmitter, nature of the neurotransmitter is a small, low molecular weight or high molecular weight neurotransmitters. That's what it signifies. Then we have what we call it as a vesicle associated membrane proteins, VAMPs. Here I have taken the diagram from the Genong's. These two diagrams are from Genong's textbook. These are the vesicle-associated membrane proteins. So now this is the synaptic vesicle. So this one, so this membrane, and this membrane is enlarged here. This is synaptic vesicle, and this synaptic vesicle is been attached with the one protein, what is called synaptobrevin, and this is also known as a V snare. V snare, that means a vesicle associated with a snare protein. Now it is known as a synaptobrevin. So now then this synaptobrevin, then we have these other molecules which are associated with the GTP, the RAP3 molecules. These are filamentous proteins, and these GTP molecules are bound and they are necessary. That's one side. Then on the other side, associated with the cell membrane, that means in this component in the this active zone, we have a syntaxin and a SNAP25. Syntaxin and SNAP25. These are also called vesicle associated proteins. So then we have intermediate, that means connectors between the V snare and T snare. The connectors between the synaptobrevin and syntaxin, we have a number of proteins. These are NSF, SNAP alpha and gamma, and MUNS18, RB-SEC1, something like that. These proteins are connecting proteins for synaptobrevin and syntaxin. So these are all put them together as a vesicle-associated membrane proteins. So I will talk about these vesicle-associated membrane proteins when I talk about the transmitter vesicular movement and the transmitter release, maybe in the next class. Now coming back, so this one we have covered the topic here up to the presynaptic components. The presynaptic components, if I were to, there is a loss of the myelin sheath, flattening or widening of the synaptic terminal or a terminal presynaptic terminal. Then there are defined active areas in which the density of the voltage-gated calcium channels and density of the snare proteins is more. These are called active zones. Then we have all over enhanced voltage-gated calcium channels especially. Then we have the microtubular system for the transport. We have the mitochondria for energy. We have the neurotransmitters here, neurotransmitter transporters on this presynaptic terminal, and these transporters are necessary for the transportation. And we have the enzyme systems within here to assemble or synthesize the neurotransmitters here inside. I have not shown, they are not shown here. Then we need to have the vesicles which are clear and dense, and we should have the membrane vesicle associated membrane proteins for the release or transport of the neurotransmitters. Moving on to the next component of the synapse is the synaptic cleft. If you are looking at the synaptic cleft, just imagine if a neurotransmitter is released here, and this is an extracellular space. This between this nerve terminal and this nerve terminal, it is an open space. And in this open space, if the transmitter is released, what happens to these transmitters? This transmitter is released explosively, and once it is released explosively, it moves to all components of the extracellular fluid via diffusion and Brownian movements and so on. And once it moves into the entire sea of the extracellular space surrounding these two terminals, so then effective concentration of these neurotransmitters would be less. It would be minimized. To prevent that, there are other proteins here. You can just see that this is the synaptic cleft. This synaptic cleft is not a loose thing, because we have to hold these two things together. First, these two terminals, presynaptic terminal and postsynaptic terminals, to be held together. And this presynaptic terminal and postsynaptic terminals, if they are to be held together, we need to have a protein molecules, which has the connectivity between presynaptic terminal and postsynaptic. These are called Neuroxins. So there are different types of Neuroxins. I am not going into the details of that. We have a different type of Neuroxins. The first Neuroxin is here, that is limiting. So this side and this side, limiting the synaptic space. So that means they would minimize the loss of neurotransmitters to the outside of the synaptic space. So then even within the synaptic space, these proteins, you just look at these proteins. This is this side, this is the postsynaptic side, this is the presynaptic side. These are the Neuroxin proteins. They are hanging on here, the green ones, and with these whatever the CHN and all these things, they try to restrict. These are proteins. They try to restrict the free flow of neurotransmitters, and they try to have that neurotransmitters trying to hold them together so that they sit on these receptors. These are the receptors on the postsynaptic membrane, and then the action begins. So this is what the synaptic cleft. Synaptic cleft is a space, and this space is about 20 nanometer wide, and this would have it has proteins within, which is connecting both the presynaptic and postsynaptic terminal, and these proteins have a function for limiting the free movement of the synaptic transmitter to the outside the synaptic area. So that is the purpose of that. So this is what I have put it here to look into the Neuroxins. I would maybe Neuroxin is a sort of a question which may come to you. I'm not going into the detail of Neuroxins because these are some proteins, filamentous and globular proteins. They would try to maneuver the pre and postsynaptic terminal. So the basic aspect, one, the synapse, the two things are to be held up together, one component, because any movement, because this is moving like this is going like this, that means that goes away. So that means they should be held in space at one point. The second point is within the space, the microenvironment of the synapse has to be maintained so that the neurotransmitter functions, neurotransmitter level is optimized to produce the effect. Then next comes the postsynaptic terminals. What are the various things present in the postsynaptic terminal? First and foremost is the presence of receptors. So here, the first and foremost, this dotted dashed line, this line here, this is the receptors. So these receptors, say for example, if it is acetylcholine, we have acetylcholine receptors. If it is a glutamate, we have a glutamate receptors. So there may be a different type of glutamate receptors. Let us not worry about that. But they're glutamate receptors. If it's dopamine, we have a dopamine receptors. If it is a GABA, we have a GABA receptors. Like that, for each neurotransmitter, where there is one receptor, and the association of the neurotransmitter with these receptors, that means these receptors act as a ligand, and then they open up the cation channels or anion channels depending upon the type of transmitter. So now they open up the cation and anion channels, and that brings about the changes the electrical activity on the postsynaptic membrane. So that's what it is doing. So now we have the receptors, we have the ligand-gated ion channels, because I have not shown the ion channels here, because the ion channels have to move here. This is the ligand-gated, and they themselves have an ion channel. Say for example, a nicotinic acetylcholine receptor, it itself is opens up the cation channels, or that would make the sodium channels to open, or chloride channels to open, depending upon the nature. Say for example, a glutamate, or let's say an MTA receptor, or if you are looking at the AMPA receptors and the GABA receptors. So like that, they will be working on. Then cation channels. So because there should be cation channels there, because they have to produce cation or anion channels for producing the depolarization or hyperpolarization. And the synapse here in this thing, it is folded. It is folded into several, so something like a villi. So that folding, the synaptic folds are the membrane folds that would try to increase or enhance the surface area, so that in this whatever the sac or the folds, the neurotransmitter enters in, wherein it will be effectively guarded by the Neuroxins. So that means that is a membrane folds, and then we have a neurotransmitter uh, destruction is required, because the neurotransmitter, if it sits on this all the time, that will keep depolarizing these things, and that may produce an excitotoxicity. To prevent that, we have an enzyme systems, especially for acetylcholine. It is acetylcholine storage enzyme system sitting here by the side. Immediately, it will start destroying the acetylcholine molecules, so that it limits the synaptic transmission. That's the component. In case of a glutamate, the glutamate transporters begin to work. So that means a reuptake mechanism would be facilitated, so that it is taken up by either by the glia or by the presynaptic terminal into the thing for further processing. Then to have all these things, you need energy, that is mitochondria. They are not shown here. So that means the postsynaptic terminals should have receptors for the transmitter. The synapse releases a specific receptors, and it should be associated with the ligand-gated channels. There should be abundance of the cation channels. There should be the membrane folds to enhance the surface area, and there should be enzymes for the destruction of the transmitters. There should be transporters in the presynaptic terminals or the glial cells for the transportation or decreasing the synaptic transmitter level. So the energy for maintenance of the membrane functions and the electrical activity, and even the chemical transduction, chemical to electrical transduction. So that is about the postsynaptic terminal. Then what are the functions of the synapse? It is a junction between two neurons. That's the definition we started with. One, transmission of impulses from one neuron to another. Okay, that's one important function. Then in case of a chemical synapse, it is a unidirectional transmission. That means it is going one way. You do not want the activity which is being directed to the brain should come back to the muscle. So that means a unidirectional transmission. So that means if the brain is sending, the brain do not want it back to the brain again. So like that. So then third thing is it modulates the impulse activity, especially in a chemical synapse. Chemical synapse is highly highly evolved, and it is necessary for the modulation of the impulse. So what is this modulation is all about? So that means modulate either as an excitatory or an inhibitory component. So that means either it is depolarization or hyperpolarization. That's one aspect. So facilitation and inhibition. So because of this synaptic transmission, whether there is a greater transmitter availability or a lesser transmitter availability, and gating, that is inhibition or suppression of the activity, divergence and convergence. So that means either all the activity are centered on one particular these things, synapse, so that they will come back. So that means they will try to disperse or focus to particular part. Convergence, as happens in case of a retina, or even if any impulse going upwards the target neuron, they will just go back by the many inputs come in on the target cells. Then from these target cells in the spinal cord, they will reach to the thalamus. So there will be a convergence, and all these things come and converge. And then when they go to the reticular system, there will be a divergence. So then the modulation is in terms of elimination or adding up the information. The memory, because this modulation of the impulse activity, either memory and sensitization. So that means either making the synapse sensitive so that you wake up, or you store the information or store whatever happened the last time, and again try to remember that and recall it and try to produce that. So that means we do our activity. If you are doing a motor activity, so what happens, suddenly you become so skilled that you do it without any difficulty. So that is one of the synaptic functions. The coding of the information, that means the impulse is coming from one neuron is to be changed, because depending upon the modulation, the coding and synchronization of the activity, and even many more. These are all the modulatory activity on the synapse by the chemical synapse. Then comes adaptation, plasticity, desensitization. Some of them are merged here, but they require an additional component there. So they adapt. That means they stop sending information. That is a sort of a desensitization, or that is a lesser activation, whatever we can talk about. Plasticity is the development of a new synapse, addition of a new synapse. So that is the plasticity. Desensitization or elimination, so that means decrementing the activity. So this is another function of the synapse. Then we have a parallel and serial processing. What is that? So if you are looking at even in the retina, some cells or some rods and cones, they will converge on one bipolar cell. So then this bipolar cell will already having some information, whether you are activating a particular cell type. So say for example, maybe if I'm talking about a color visibility, the blue, green, or red. So when I'm talking of these primary colored rods and cones, so then they are processed there already in the parallel manner. The serial processing is done in terms of one processing happening at the retinal level, processing happening at the ganglion cell level, processing happening at the lateral geniculate body, if I were to talk about the visual system. If I were to talk about the spinal cord, that the peripheral inputs, if I were to talk about anything coming from the skin, it is to be processed in the spinal cord. They will try to segregate at one place. From here, it is taken up further. So that means a parallel and serial processing, and especially certain sensory modalities are processed in the thalamus. So that means that is how the processing take place. Each level there is a processing. Then in case of some synchronized activity, especially in the hippocampus and in the medulla, we have this synchronization, and the pacemaking activity is very much required to produce an effective response. So that means these are 5 or 6 functions. 6 functions I have listed here of a synapse. The primary one is the transmission of an impulse. Rather, I would say a transduction of an impulse from electrical to chemical to electrical, chemical, electrical transduction. That is one function. Then trying to modulate the impulse activity, whether to enhance or decrease, or whether to gate, whether to diverge or to converge, whether to eliminate, whether to then it will try to remember, and already sensitized, and it will help in coding of the information for further transmission. So that is how the functions of the synapse work. So I have already listed all of them. Some these allow us for processing of the information, and even for synchronization of the information. Now, let us look into the types of chemical synapse. So now here I have made a simple diagram, the neuronal synapses. This is one neuron and there is another neuron. And now this neuron would be making a synaptic contact. It's made a synaptic contact here on the soma. This is soma of the other things. And some of them are making a contact with the dendrite. So that means they have a synaptic contacts. This is one neuron to another neuron, neuronal synapse. You are already familiar with a neuron and a muscle contacts, neuromuscular. That neuromuscular junction you have studied. So that means from one alpha motor neuron to the skeletal muscle here. So this is one neuromuscular junction or neuromuscular. Neuroglandular, say for example, to the salivary gland, whatever the autonomic inputs, the parasympathetic nerves, they supply the salivary glands. These gland cells, they will be activated. Maybe salivary, maybe parietal cells of the stomach, or maybe exocrine glandular structure of the pancreas by the vagus. So this is a neuron to gland, neuron to muscle, and neuron to neuron. So that means neuronal, neuromuscular, neuroglandular. Now, if you are looking at the neuronal synapse, there are primarily or most commonly three types of synapses are there. Neuronal synapses. Common. These common are exodendritic. This is a dendron of this nerve fiber. This is a nerve fiber. This is the dendron. This is the dendritic spine, and this dendritic spine and the axon of another neuron, they are making contact. This is exodendritic synapse. See, for example, a transmission is taking in this direction. So now here is another, because this is the dendritic spine or dendritic knob. Here on the dendritic surface here, there is no knob there. So this is also exodendritic. Now this axon is making a contact with the soma. This is exosomatic. And another axon here, it's making a contact with the axon of this cell. This is exoaxonal. That is what I have mentioned: exodendritic, exosomatic, and exoaxonal. Less common are sometimes the two dendrites may talk to each other maybe for sensing purposes. Dendrodendritic. There is a spelling mistake here. Maybe I will change that spelling mistakes. Exodendrodendritic. Dendrodendritic. This is one is quite often seen, especially in certain areas in the spinal cord, in the cerebellum, in the cerebrum. Dendroaxonal difficult, because dendrite is a reception, is difficult proposition. Dendrosomatic is possible because the dendron of one nerve may have a contact with the soma of the other, so that is possible. Somosomatic, so that means from body to the cell body of this to another cell body is a possibility. So what is not possible? Dendroaxonal is not possible, but dendrodendritic, dendrosomatic, somosomatic are also possibilities, but they are less common. These are the very frequent or common types of synapse, neuronal synapse. Here I am trying to give you an example. This is the picture taken from the cerebellum from Genong. Okay, I just explain you here. This is one Purkinje cell, and these beaks are the dendrites, and this one is axon. This is a basket cell. This is a dendrite, and this is a axon. You just see here This is making contact here. What is this? This is axon of the granule cell, axon of the granule cell. So axon of the granule cell make the dendrites. The beak is, this part is the dendrite, exodendritic. Or if you are looking at, this is the axon from the axon coming from the olivary nucleus, the climbing fiber, and making contact with the dendrite, exodendritic. Sometimes it will make a contact. Here it has made a basket cell making contact with the soma, exosomatic. So this is the axon, exosomatic. So these are exosomatic. So now here you come. So these are dendritic, exodendritic. This is a exosomatic, and this is exosomatic. So like that we have, I have given just taken examples, but in the Purkinje cells, the basket cells and the stellate cells, they are there, and they may have dendritic connections also. They will try to contact. So that is one possibility here in this these things. This is the diagram just to explain that such a type of synapses do exist. Maybe in some pictures I would show you the real. Now, what are the sequence of events happening during synaptic transmission? One, I have made the numbers here: 1, 2, 3, 4, 5, 6, 7, 8. I have made 8 events. The first event is the arrival of the impulse from the presynaptic terminal. Arrival of the impulse. Number 2, the arrival of impulse changes the voltage, or it depolarizes this membrane. So change in the voltage results or opens the calcium channels. So this calcium channel opens. So especially this is a voltage gated calcium channels, opening of voltage gated calcium channel. Now opening of this channel makes the calcium to enter in. So that means calcium influx. Now what this calcium does, now this calcium is used or triggers the vesicle associated membrane proteins, VAMPs I was trying to talk about, that would activate the vesicle associated membrane proteins, synaptobravin. So now the synaptobravins trying to move the vesicle to the docking system. That means vesicle number 5 is the movement of the vesicle by these vesicle associated membrane proteins and intermediate. That means this is a V snare and a T snare and intermediate proteins. They all put together, they move this vesicle to the docking site, the docking protein. Now, then 6th is the kiss and run. For the exocytosis, so what happens here? This comes here, it touches this membrane, and just it runs away. It runs away back. Maybe in the next class I will talk in detail a little bit more. So that means a kiss and run. That means exocytosis. Here, it touches this membrane, it releases the neurotransmitters and runs away, closes this thing, and carries this empty vesicle. Now, the transmitter released is here in this synaptic cleft. Now, it gets attached with the receptor, the neurotransmitter and receptor interaction. That is the 7th. And then neurotransmitter receptor interaction opens up the channels, ion channels, or ligand-gated ion channels. And this opening of the ligand-gated ion channels either produce the excitatory postsynaptic potentials or inhibitory postsynaptic potentials and bring about the transmission of the impulse. These are the sequence of event happening in the neurotransmission. Well, I have covered this today. I have covered up to 6 events for this part of the synapse. Now, I am trying to give you some assignments. I want you write it down in 2 to 3 sentences for each of them, because what I would do, in the evening I will upload this thing in the YouTube lecture so that you can revise it. You can revise it. Maybe it will take about 1 or 2 hours for me to process. Okay, so now what you do is you work on this assignment. So you define synapse, or what is synapse, name the parts of the synapse, write the differences between electrical synapse and a chemical synapse, describe the advantages and disadvantages of an electrical synapse, name the cellular components of the presynaptic terminal, what are Neuroxins and their functions, components of the postsynaptic membrane, classify the types of a neuronal synapse, list the events happening during synaptic transmission, and list the functions of the synapse. These are the 10 questions. Try to make them and write in your assignment book so that would make you to understand this particular topic in a small bit. So that means it covers everything, and try to work on it. Okay, these are some of the references. That means Kendall's Neuroscience, the guidance textbook of medical physiology, Genong's review of medical physiology, and Samson Wright were the basis for my, and even many more some articles. Okay, I do not want to bug you with the entire references. So the next class, what we will do, we will continue with the part 2 of the synapse, because I did not touch upon this vesicular transport, and its recycling, the neurotransmitters, and the properties of the synapse, which I would take in my next class. Okay, so